Okay, just a reminder, the Calvin cycle, Rubisco is the enzyme. Here's the slide from before. So ribulose 1,5 bisphosphate is the chemical, right? With water, with CO2. In the presence of Rubisco, ribulose 1,5 bisphosphate carbylase, oxygenase, ACE is the enzyme. So Rubisco is the enzyme, ribulose 1,5 bisphosphate is the chemical compound. Put those together, and that gives us the beginning of what will, through the Calvin cycle, be our glucose molecule. Just a reminder that one is the enzyme because it has an ACE on the end, and one is the chemical compound. Okay, electron transport chain. You are looking at this again from the thylakoid membrane in photosynthesis and comparing it to the mitochondrial membrane in respiration. And it really is quite similar. You're looking at photosynthesis up here, right? Here's our light reaction. First one, photosystem 2. Here it goes, photosystem 1. Then we're moving all of those hydrogen ions, and then through ATP synthase, you are getting ATP, and that is fueling the Calvin cycle to make your glucose. Mitochondrial respiration, very similar. Here's our electron transport chain pushing our hydrogens. Our hydrogens come out, ATP synthase, and it runs the Krebs cycle. In both, the only way for proteins to move back down their gradient is through ATP synthase, which is sort of our hydroelectric water wheel, turning ADP into ATP. Okay, energy carriers. This is our NADPH and NADH in animals, and these are our little cars, carriers, however you would think of them. While they're chemically similar, they do deliver two different routes. So NADPH, anabolic reactions. Its destination is the Calvin cycle. Okay, so it's made here in the photosystem 1 and photosystem 2, and then we're using that NADPH to run the Calvin cycle to ultimately make us glucose. Then compare that to the NAD and NADH and FAD here in the electron transport chain. Same thing, we're using it to carry the electrons. It is the catabolic reaction. Its destination is the electron transport chain here with ATP synthase. Okay, the G words, right? Glycogenesis, the other G words, which are present in plants and which are only in animals. So while plants use starch as their storage, animals use glycogen. Therefore, any term specifically related to glycogen, like glycogenolysis, is specific to animals. Insulin facilitates glucose, glycogenesis, glycolysis. Glycogen and epinephrine trigger glycogenolysis and gluconeogenesis. So we have a lot of papers on that, and we talked about that a lot, all of the G words. Okay, plant growth regulators. Especially gibberellins. Okay, so these are going to promote stem elongation, seed germination, flowering. They are crucial for seed dormancy. How are these doing it? Let's look at both of those things that they do. I'm sorry for the snoring pug. So one of the things they do is they promote stem elongation and seed germination by releasing the cell wall. Okay, so the mitosis and meiosis can only do so much because remember, plants have that rigid cell wall. So one of the things that your gibberellins do and how they promote stem elongation and germination is that they break that, they don't really break it, they just loosen the cell wall. So the cell wall can get bigger and the plants can also get bigger. Okay, let's talk about apical dominans and auxins. So auxins promote cell division. So you can see how auxins and gibberellins are going to work together. Gibberellins are going to loosen that cell wall, and auxins are going to promote cell division. They also promote root initiation and apical dominance, and the apical dominance is related to gravity. So if you look at how an auxin works, its receptors direct growth downward in response to pressure from gravity, and that's how it knows to grow up, related to the sun and related to opposing forces of gravity. And here it is the sun, right? When the sun is directly overhead, it gets evenly distributed on all sides. So again, another thing auxins do is they prevent premature leaf drop and encourage fruit development without fertilization. So that is going to be things like seedless grapes. Okay, auxins maintain that apical dominance by inhibiting the growth of lateral buds. So when you cut off the top, so here's your lateral bud, if you cut that off, then you're going to get branching out. So if you want to have a plant that's short and goes out, you're going to cut the bud off, and that's going to, when the source of the auxin is removed here at the top, the lateral buds begin to grow because the signal is actually inhibitory, and it's going to start moving that to the buds, and you're going to get a short plant that is growing outwards. Okay, let's compare the signals of insulin in animals to auxin in plants. So both of those have a calcium secondary pathway. Do you remember insulin? Here is the insulin pathway, and we had calcium binding. We had a calcium voltage gated calcium channel that came in and bound and released our insulin in the beta cells of our pancreas. Do you remember that? The voltage gated calcium channels. Very similar here. We have a voltage gated calcium channel related to auxin. Cytokinins. So these stimulate cell division and they delay aging in leaves. And again, they work with auxin to regulate organ formation. Ethylene is a gaseous hormone that triggers the fruit ripening. It promotes shedding of leaves and influences stress response. So your ethylene response is going to be, very often they will pick fruit when it is not ripe. They will put it on a truck and then they will gas it with ethylene at the destination so that it doesn't get squishy and rot on the truck ride over. Okay, next, abscisic acid. This is a growth inhibitor. It promotes dormancy and assists in tolerance to environmental stress, and it's the main way in which we close the stomata. It also is what will actually abscise or remove the leaves, right? Okay, so let's talk about this abscisic acid pathway in the stomata. It is, again, a calcium secondary messenger. So what's going to happen, stay with me, you are going to get abscisic acid that's going to open a calcium channel. That calcium channel is actually going to open up all of these other, like chlorine and potassium. The chlorine and potassium are going to move out. When the chlorine and the potassium move out, what's water going to do? To remember osmosis, water is also going to move out. That's going to shrink our stomata. Okay? So under water stress, your abscisic acid is going to open a calcium channel and all the other potassium, and this causes the water to leave and deflates the guard cell. Okay, so that is how our calcium channel is used in stomatal closure. Okay, and here is sort of how it happens naturally. When there's blue light, which is in the evening, it closes. So blue light stimulates your ATPase proton pump, providing the membrane potential needed for guard cells to import potassium and sugar, and as a result, water enters by osmosis. Okay, so it enters with the blue light trigger and the potassium. It exits typically when we are talking about abscisic acid, the calcium channel. Okay, and there's both of those. Okay. Absorption phase. So some more transporters. Okay, here we have SIGLT, sodium glucose transporter, GLUT, glucose transporter, sodium potassium pump. How they enter the bloodstream. And again, we're comparing their calcium channels. So we've got receptors looking for hormones like abscisic acid. Then there's the secondary messengers of calcium ions. And then that abscisic acid during drought or times with no water will actually trigger the closing of the stomata. We just had that in the last slide. Okay, but now remember, here's SIGLT, sodium glucose transport. The sodium flows down. Okay, that is a symporter because both of them are coming in together. Glucose, sodium, this is a symporter. Okay, the sodium flows down its gradient into the cell and it brings glucose with it. It's like, hey, let's go, man. We're going to go into the cell. GLUT has an intercellular glucose, drives its facilitated diffusion, and then it moves. GLUT is going to be the one down here that's going to move it into the bloodstream. And again, our sodium potassium pump is an antiporter, right? Sodium comes out, potassium goes in. Again, the pug is snoring, I'm sorry. Compare and contrast this with the plant growth regulator, specifically the calcium channel with abscisic acid and the pancreas's release of insulin. So again, here's our abscisic acid voltage gated calcium channel. Abscisic acid triggers our voltage gated calcium channel. Calcium comes in, and we have all these chlorine and potassium that go out, water goes with it, and that is our voltage gated calcium channel. It promotes the hydrogen peroxide production, the leaving and activation of these channels. Okay. And when you compare that to the pancreas, right? Our pancreas, I have a slide of that on the old slide set, but I think it'll come up. Again, if it doesn't, I will bring that slide up again at the end. But we again have the calcium, a voltage gated calcium channel in our pancreas, and that's going to release the insulin. Mendelian genetics. So this is a trip back to the beginning of the semester, right? This is just a reminder how these work. There's not a lot of questions. There's not essay questions. These are just multiple choice, but just a reminder to look over it. Heterozygous, right? Here is our big Y, little y, big Y, little y, and then we're going to get a 1 to 2 to 1 or a 3 to 1 phenotypic ratio. Okay, so 3 to 1 phenotypic. Let me move 3 to 1 phenotypic, 1 to 2 to 1 genotypic. This is Mendel's law of segregation, which states that allele pairs separate during gamete formation. Little meiosis there. Okay, also a reminder about incomplete versus co-dominance. Incomplete dominance is usually, again, I like to think of it like a red and a white makes pink because there's just not enough protein here to make red. So red and white make pink, like blending of paint. Like there's just not enough of this, so it makes a pink color. That's incomplete dominance. Creates sort of an intermediate. Codominance is both of them are exhibiting, they're both showing. So the example here in the flower would be they're red and white, and from far away it looks pink. Or fur, that's black and white, and from far away it looks brown. The best example of codominance that's the most obvious is your blood type. Like if you're AB, you exhibit signs of being A and signs of being B. So that is the difference between incomplete dominance and co-dominance. Genetic expression. How can the environment affect the expression of a plant's genotype? So this can definitely be like Himalayan rabbits where we have the cold. Sometimes it is affected by temperature. Sometimes it is affected. So here's temperature. It can be affected by temperature. It can be affected by light, depending on how much light something gets. Remember when we talked about variegation and the white parts of the plant? Sometimes that will be, you will see that if the plant is getting enough light. But if the plant is not getting enough light, it will stop producing that variegation regardless of the genetics because it's not getting enough light. Light also tends to trigger genes for compact growth and highly efficient photosynthesis. Low light induces stem elongation, things like that. Sometimes you get fruit ripening, fruit shape related to light intensity or temperature, or sometimes you will get based on how crowded insects are. They will have wings or not wings. All of those are interesting genetic expressions related to environment. It is really a fascinating sort of evo-devo section of genetics. Active transport. We're back to transports again. Okay, I only have 7 more, so we're just going to push through and make this one a little bit longer. Okay, why do plants' roots require active transport to pull minerals from the soil, even when the concentration is higher inside the root? Basically, it requires you to get as much, there is ATP here because you need to get as much from the soil as you can because those are your nutrients and you don't know when they're always going to be there. So they use active transport to pull minerals from the soil, even when the concentrations are higher, even when they have to move against their gradient because they need to store those nutrients. So it can acquire nutrients like phosphorus, potassium, and nitrogen that are often scarce in the soil or concentrated inside the root, which then from passive diffusion can take it from there. Homeostasis. How does the plasma membrane use passive transport osmosis to maintain turgor pressure in plants? So the plasma membrane maintains turgor pressure through passive osmosis. So this is our whole passive osmosis all the way up from the root, the little tiny root hairs, all the way xylem mostly and phloem all the way up to the top. This allows water to move down its concentration gradient into the cell's central vacuole all the way up here. Okay, here's our turgor pressure. Here's low turgor pressure, here's high turgor pressure, and it just continues to keep that. Okay, again, our secondary messenger of calcium ions, looking at our abscisic acid, controlling the stomata closure. Metabolic regulation. How does feedback inhibition allow the cell to shut off metabolic pathways? So essentially, the end product is the path to shut off, right? It attaches to that enzyme. So feedback inhibition shuts down metabolic pathways by having the final product, for example, the amino acid that's being made, in this situation, actually bind to the enzyme to shut it off. So it can change its shape so it can either catalyze or stop the reaction. And this, again, is to keep it from making and using energy that it does not need. We don't want to make things that we don't need. If you're not drinking milk, there's no reason for your body to be making a bunch of lactase. It's unnecessary, so your body does not need to do it. When are red blood cells placed in a hypertonic solution? How does the membrane facilitate this movement? So water, remember, is always going to rush out to dilute things. So in this case, we have a hypotonic solution. So water is going to rush in. Here we have a hypertonic solution. Water is going to rush out. Just a little reminder. Cellular junctions, gap junctions, plasmodesmata. So gap junctions in the animal versus plasmodesmata in the plant allow direct cellular communication by physically connecting the cells that are next to each other. Gap junctions here in the animal are protein-lined pores that go in between versus plasmodesmata are membrane-lined tunnels that traverse the cell wall. Because remember, we have a hard cell wall here, and these are not a hard cell wall, but we still have gap junctions in between them. And passive transport, molecules move down their concentration gradient without using ATP. So this is just passive transport. You can just have a regular old passive transport that is simple diffusion. You can have a transporter that's sort of passively doing it. But when we think of passive transport, we really just think about moving down the concentration gradient. When we look at facilitated diffusion, that's when we have glut, Siegelt, sodium potassium pumps, aquaporons. Right, here's a uniport. It's just moving one thing down. This would be like glut, glucose transporter, versus a symport, which would be a Siegelt glucose sodium transporter. And you have an antiport, which is in my eye like the sodium potassium pump, but this is giving you an ATP ADP example. Okay, that is it.